Hello, I am Dr. Özlem Yalçın Çapan from Tekirdağ Namık Kemal University. The title of my presentation today is Evaluating the Performance of Bioinformatic Tools for Analyzing CDKL5 Gene Variants. The CDKL5 gene is located on X chromosome and codes for a serine threonine kinase protein that is involved in various signaling pathways within neurons. So this protein consists of a catalytic domain and a long C-terminal extension. The catalytic domain is located in the N-terminus and it is responsible for the catalytic activity of the protein, while the C-terminal extension regulates the catalytic activity and subnuclear localization of the protein. This protein is vital for the normal neurological development, and its proper functioning is essential for maintaining cognitive abilities and motor functions. Unfortunately, mutations in the CDKL5 gene are associated with a range of severe neurological and developmental disorders, such as the CDKL5 deficiency disorder, CDD, developmental and epileptic encephalopathy, DEE, and the Rett syndrome. All these are rare genetic disorders and they manifest early in life, often within the first few months, and are characterized by severe epilepsy, developmental delays, intellectual disabilities, and motor dysfunction. Today, more than 200 missense variants have been identified in the CDKL5 gene in the ClinVar database. However, some of these variants have benign effects on protein structure and function. So identification of the true causative variant for these disorders is crucial for achieving accurate diagnosis and implementing precise treatment for the patients. So here we are using the American College of Medical Genetics and Genomics, ACMG standards and guidelines for the interpretation and to detect the correct variant which is causing the disease. So these guidelines suggest using multiple software approach for variant interpretation, but does not specify the exact number or types of the algorithm. Today, there are more than 100 bioinformatic tools developed on different principles, and we use them to assess the pathogenicity of the genetic variants. Unfortunately, these tools have a success rate ranging from 60% to 80%, and each tool and its underlying algorithm has its own advantages and limitations. Therefore, selecting the appropriate bioinformatic tool is crucial for accurate variant interpretation. So in this study, our aim is to evaluate the performance of commonly used three commonly used bioinformatic tools, which are SIFT4G, MetaLR, and MutPred2 for the analysis of CDKL5 variants. As a method, first of all, we selected 94 pathogenic or likely pathogenic and 93 benign and likely benign missense variants in the CDKL5 gene, which are associated with DEE, CDD, and the Rett syndrome in the ClinVar database. And secondly, we obtained the prediction scores for these bioinformatic tools for the missense variants in this gene. And then we evaluated the performance of these bioinformatic tools. So we used different calculations here. We calculated the sensitivity, specificity, accuracy, and also Matthews correlation coefficient scores, as you can see the equations in the slide. So as a result, first of all, I want to introduce you to these bioinformatic tools. The first one is SIFT4G, the other is MetaLR, and then MutPred2. SIFT4G integrates information from protein sequences, alignments, and evolutionary conservation to predict the impact of each possible amino acid substitution. And here we have a score ranging from 0 to 1. And if we have a value between 0 and 0.05, then it is predicted to affect the protein function. So MetaLR, on the other hand, is a meta predictor and it incorporates 10 scores such as Swift polyphenol mutation tester, and also the maximum frequency observed in the 10,000 genomes populations. So for meta-LR, the score can range from 0 to 1, and if we have higher values, this means it is more likely to be delicious. Multipred2, on the other hand, uses machine learning techniques, specifically neuronal networks, to predict the functional impact of amino acid substitutions. This model is trained on a large dataset of known pathogenic and benign variants. Multipred2 incorporates a wide variety of features, including sequence conservation, protein structure, functional annotations, and also physicochemical properties of the amino acids. Here, if we have a score over than 0.5, then this will suggest the pathogenicity. So we also evaluated the performance of bioinformatic tools, first of all, by carrying out a ROC analysis. So the ROC curves, as you can see, is a graphical representation commonly used in binary classification tasks to assess the performance of the predicted models. And we also calculated the area under the curve AUC of these ROC curves because it quantifies the overall performance of the model. And if we have a higher AUC scores, this indicates a better discrimination ability. In general, we can say that if we have a score of AUC between 0.7 to 0.8, this is acceptable. If we have a score between 0.8 to 0.9, then this is considered good. But if we have a higher score than 0.9, this indicates an excellent classifier. So as you can see, the figure here in the slide, Multipred2 and the meta-LR actually have higher AUC scores over than 0.9, and this indicates that these tools are excellent classifiers. We also calculated the Matthews correlation coefficient scores. This is a statistical measure commonly used in binary classification again. And as you can see here, Multipred2 specifically has a higher score for MCC, again indicating a more reliable classifier actually. We also calculated sensitivity and specificity, which are key metrics used to evaluate the performance of bioinformatic tools. And sensitivity measures the proportion of the actual pathogenic variants that are correctly identified by the tool. And if we have a high sensitivity, this means that the tool successfully identifies most of the pathogenic variant. And as you can see here, meta-LR and Multipred2 have higher scores for the sensitivity, higher than 90%. And specificity measures the proportion of actual benign variants that are correctly identified by the tool. And if we have a high specificity score, this means that this tool successfully identifies most of the benign variants. As you can see in the table, Multipred has a high specificity score, more than 90%. We also calculated accuracy, which measures the proportion of correct predictions out of all predictions made. Accuracy represents the degree to which the tool correctly classifies both positive and the negative cases. However, it doesn't distinguish between the types of errors made. But as you can see here, for the accuracy scores, again, we have a very high score for the Multipred2 tool. We also calculated the pathogenic cutoff scores, which indicate the optimal cutoff scores for each tool to evaluate the missense variants in the CDKL5 gene. So as a summary and a discussion, in this study, we aim to evaluate the performance of three widely used bioinformatic tools, SIFT4G, meta-LR, and Multipred2, in the analysis of the CDKL5 gene variants. So in this study, the evaluation criteria included accuracy, sensitivity, and specificity, which are crucial metrics for determining the reliability and effectiveness of these tools in predicting the pathogenicity of genetic variants. So here, our results demonstrate a clear distinction in the performance of the three tools, with Multipred2 emerging as the most effective in analyzing the CDKL5 variants. Specifically, we can say that Multipred2 achieved the highest accuracy with 96%, sensitivity with the 98%, and specificity 95%. So in these metrics indicate that MutSpread2 not only correctly identifies the majority of true positive and true negative variants, but also minimizes the number of false positives and false negative ones. While, by the way, SIFT4G and MetaLR, they are commonly used and valuable tools in the bioinformatics community, their performance in the study was notably lower compared to the MutSpread2. As a conclusion, the evolution of SIT4G, MetaLR, and MutSpread2 underscores the importance of selecting the right bioinformatic tool for genetic variant analysis. Thank you for listening.